The ear is the organ of hearing and balance. One of the very advanced and sensitive organs of the human body, the ear detects, transmits, and transduces sound to the brain and maintains a sense of balance. The ear is divided into three major areas: external ear, middle ear, and internal ear. The external ear consists of the auricle and the external acoustic meatus. The auricle, or pinna, is composed of elastic cartilage covered with thin skin and occasional hair. The function of the auricle is to direct sound waves into the external acoustic meatus. The external acoustic meatus, or auditory canal, is a short curved tube that extends from the auricle to the eardrum. The entire canal is lined with skin-bearing hairs, sebaceous glands, and modified apocrine sweat glands called ceruminous glands. These glands secrete yellow-brown waxy cerumen, or earwax, which provides a sticky trap for foreign bodies and repels insects. The middle ear consists of ossicles called malleus, incus, and stapes. The ossicles amplify the sound force. They increase the sound force at the oval window, and smaller surface area at the oval window. The air in the middle ear is continuous with the air in the nasal cavity via the eustachian tube. This tube is usually closed by a valve. The inner ear is entirely enclosed within the temporal bone. It has two separate regions, the cochlea and vestibule, which are responsible for hearing and balance respectively. The neural signals from the two regions of the inner ear are relayed to the brainstem through separate fiber bundles, but which run together as the vestibulocochlear nerve. Sound information is transmitted from the middle ear to the inner ear via the stapes attachment to the oval window, which is a membrane at the beginning of the cochlea. As the tympanic membrane vibrates from sound waves, the ossicles amplify that vibration, and then the oval window moves with the same vibrations. As vibrations of the tympanic membrane are transmitted through the ossicles, a wave is created within the fluid in the cochlea that displaces sections of the cochlear partition. The tube running from the oval to the round window in the cochlea is separated into two spaces. From the oval window to the tip of the cochlea, the tube is referred to as the scala vestibuli, and from the tip of the cochlea back to the round window, it is the scala tympani. The two spaces are on either side of the scala media, or also called cochlear duct, which is the space that contains the organ of Corti. The organ of Corti transduces sound into the neural signal. The organ of Corti contains hair cells with stereocilia on their apical membrane. The stereocilia bend in response to movement of the basilar membrane relative to the partially fixed tectorial membrane. Depending on which direction the stereocilia bend, they open or close ion channels, leading to signal changes in the cochlear nerve. As a standing wave is set up within the scala vestibuli and scala tympani in response to movement at the oval window, the basilar membrane responds by moving at a specific spot, depending on the frequency of the standing wave. The cochlea encodes auditory stimuli based on frequency between 20 to 20,000 Hz, the range of human hearing. Higher frequencies cause the basilar membrane close to the base of the cochlea to move, and lower frequencies cause the basilar membrane closer to the tip of the cochlea to move. Loudness is determined by the number of hair cells in an area that are stimulated. A louder sound produces greater displacement of the basilar membrane, leading to a greater number of stereocilia responding. The information sensed by the spiral organ of the cochlea is transmitted into the brain via the vestibulocochlear nerve to nuclei in the pons. At the level of the pons, and related connections in the midbrain, some processing of auditory information occurs, including identifying where a sound is coming from, and responding to loud noises. From the pons and midbrain, fibers carrying auditory information project to the thalamus, and then from there to the primary auditory cortex of the temporal lobe. It is here that the conscious perception of sound is located. Similar to the cochlea, the vestibular structures use hair cells with stereocilia to detect movement of fluid, in this case, in response to changes in head position, or acceleration. Detection of head position when the body is stationary is termed static equilibrium. The information for static equilibrium comes from the utricle and saccule. Dynamic equilibrium is the perception of acceleration. Information for dynamic equilibrium can come from the utricle and saccule, which detect linear acceleration, and or the semicircular canals, which detect angular acceleration. The neural signals generated from the vestibule are transmitted to the brainstem and cerebellum from sensory neurons in the vestibular ganglion. The saccule and utricle each contain a sense organ called macula, where stereocilia and their supporting cells are found. These maculae are oriented 90 degrees to one another, so that they respond to positions in different planes. The organs can respond to changes in position and acceleration because the tip of their stereocilia project into a dense otolithic membrane made up of a mixture containing granules of calcium and protein called otoliths, which gives rise to their common name, the otolithic organs. When the otolithic organs move, gravity causes the dense otolithic membrane to move relative to the less dense cell layer beneath the stereocilia. This causes the stereocilia to bend, initiating action potentials in the vestibular nerve fibers that innervate them. Bundles of stereocilia are arranged in various directions, so that any direction of inclination will depolarize a subset of the hair cells, and hyperpolarize a corresponding subset of sensory neurons. The semicircular canals are 3 ring-like extensions from the vestibule. One is oriented in the horizontal plane, and 2 are in the vertical plane. The vertical canals are 45 degrees off of the sagittal plane. One is anterior, and one is posterior. At the base of each semicircular canal, where it meets with the vestibule, is an enlarged region known as the ampulla, which contains a hair cell-containing structure called the crista ampullaris that responds to rotational movement. The stereocilia of the hair cells extend into the cupula, a membrane that attaches to the top of the ampulla. When the head rotates in a plane parallel to the semicircular canal, the fluid in the canal does not move as quickly as the head is moving. This pushes the cupula in the opposite direction, deflecting the stereocilia and creating a nerve impulse. Considering the semicircular canals on either side of the head, 3 orthogonal planes are defined: the horizontal plane with both horizontal canals, and 2 vertical planes 90 degrees to each other with the anterior canal from one side, and the posterior canal from the other. In each pair, deflection of the cupula on one side of the body causes depolarization of the hair cells, while the same movement causes hyperpolarization of the hair cells on the other side of the body. Information from the vestibular apparatus projects to a wide range of structures in the brain. The information enters the brain via the vestibulocochlear nerve, where it synapses at vestibular nuclei in the pons, and medulla. From here, information travels to a number of other regions to stimulate reflexes and develop conscious awareness of position and movement. If you like this video, please hit the like button below and subscribe to my channel to see more videos in future. Thank you for watching. Bye.